very good microsurgical lab, and I came and I established the Department of Transplant Immunology and Cell Therapies. And starting from 2004, here is actually where we did all our research. In this department, I aimed to develop only on translational approaches, meaning that I only followed things which had the chance someday to be of some help to patients, and not only to discover things in vitro or in vivo in animals. And in this direction, we had two main aims. So first was to follow the tolerant strategies in solid organ transplantation, what I have began in Germany, and of course this was not in the end because we did not have the time. I left a very solid protocol that could be eventually applied in the clinic, and I concentrated on mitomycin and dendritic cells and T regulatory cells. And therapeutic neoangiogenesis, this is a thing which came later, and here we actually tried to develop endothelial progenitor cells which could induce neoangiogenesis into ischemic tissues. All of these subjects were based on a financial backbone of about 831,000 euros, which we earned in this lab during 2004 and 2010 actually was the last year when we got financing, and from that on everything died from this point of view. So taking mitomycin from bench to bedside, okay, this was a collective effort, an international effort, in which we as coordinators implied the University of Heidelberg, the University of Cluj-Napoca, and the Clinical Institute Fundeng. So what we actually did here is, of course, we had the results in the rats, but we were wondering what is happening in humans. So we started to do the same thing and see whether we have the same proof of concept in humans. So we developed the dendritic cells and then incubated the dendritic cells with the same allogeneic T cells and see in humans whether whether the suppression was happening or not. And to our surprise, suppression was happening. It was not that strong as in animals, but a very significant suppression in comparison with controls was there and was giving us the light and the power to go further on. Moreover, when we tried to take these cells, which were already suppressed, and incubate them with fresh dendritic cells, which are very powerful from a stimulating point of view, we found out that those cells cannot be restimulated. So once those cells were suppressed, they were staying in that actually state, and this was very important for the experiments that were about to come. Mitomycin was not toxic to human dendritic cells, so this is very good. But we wanted to see why is actually suppression happening. And one thing we discovered is that during suppression, actually those T cells, because of the mitomycin-secreting dendritic cells, are transforming themselves into T regulatory cells. This is a special subpopulation of T cells that exists normally in our bodies, and it's responsible to not allow autoimmune disease to happen, but they can also be induced in vitro and in vivo. So these are the controls, and once we took out cells from these cultures, you could see that those cells were getting very highly positive for two markers which are fundamental for T regulatory cells, which is CD4 and FOXP3. This is a 14 protein B3, which has been described as a fingerprint of this type of cells. And to my surprise, years later in Heidelberg, the co-workers in this project found out that intolerant hearts of rats receiving mitomycin-secreted dendritic cells, they had lots of T regulatory cells compared with the non-treated hearts. You can see the T regulatory cells are the ones colored in brown, and you can see a very high significance already shown in this graphic. Okay, but were these T cells, T regulatory cells, were these cells suppressive, so we could use them in a transplantation scenario? Yes, they were. What we did is we took out those cells and combined them with fresh mixed lymphocyte reactions, which were very important from the statistical point of view. And we have found out, again, that a significant suppression of proliferation was taking place. And this was due to our Tregs, and this suppression was actually found out to be happening into a concentration-dependent manner. This paper has brought us the top 10 rated research at the European Society of Organ Transplantation Congress in Paris in 2009. Top 10 rated research at this Congress, let me tell you, this means a lot, because we were rated top 10 within 890 posters from the same category, meaning experimental immunology. And later on, follow up the paper which we published together with the group in Heidelberg regarding mitomycin C treatment of antigen-presenting cells. And this is a review appeared in the Human Immunology, and it contains actually all the preliminary data I've been showing you until now. I did it in my postdoctoral years. And just to show you another very important thing, which I think has to do a lot with persistence and a lot with developing further a concept which I developed in Heidelberg. I came back to Timișoara, I did everything in humans, but in Heidelberg the people follow this concept and try to do the same thing in autoimmune disease. Multiple sclerosis, for example, everyone knows about this disease. It is a terrifying disease, it cripples the people, and it cannot be stopped for the time being. And what is happening with this is actually an autoimmune reaction to a basic myelin protein. This is a protein into the brain of patients. And once you can find out which are the cells which are killing this protein, probably we have a cure for multiple sclerosis. So they try to apply the mitomycin concept into models of multiple sclerosis. This is the experimental autoimmune encephalomyelitis mouse model, and this mouse has been preconditioned with cells with mitomycin, and this mouse has been left. These mice developed the disease spontaneously, if not treated, okay? And this mouse was left alone, while this mouse, before he developed the disease, he was pre-treated with these cells combined with mitomycin, and of course the disease could be prevented. Moreover, ongoing multiple sclerosis in mice was treated using the same concept. So this mitomycin treated, they are actually mitomycin treated and treated cells which are specific for basic myelin proteins, so they go there and actually they block the development of the autoimmune reaction, so the animals can regain their normal habitus. And these two videos were very significant and very important, because actually in 2007 we had the first clinical case where we applied this technology. This was a 6-year-old patient with acute lymphatic leukemia, which already has received 2 times the haploidentical stem cell transplant from mother and father, and she acutely rejected this transplant after 40 to 50 days. She was at that stage reconditioned with several monoclonal antibody therapies, and received another, the 3rd haploidentical stem cell transplant from the father. Unfortunately, this began to be rejected again, and this is where the mitomycin came into place, because they have isolated from the father an enriched ratio of cells which were incubated with mitomycin, and the patient received 2 times, 1 time 10 to the 9 cells, and then in the second treatment, 2 times 10 to the 9 cells, and she was able to hold the graft for 200 days, meaning that she could reject the blood cancer that was happening in her blood. Unfortunately, she succumbed after about half a year later because of problems related to a lung infection. So in 2009, we have the proof of concept, and we were able to patent this technology, and this technology is now patented. I'm one of the co-authors. But we have proved the concept in rats. We have proved the concept in mice. Okay, we have seen that something is going on into humans. We had the clinical proof of concept. But in between mice and humans, there was still a big evolutionary gap. And for this, the Department of Transplant Immunology and Cell Therapies in Timișoara took the task to close this gap and see what is happening in transplantation. And we took the pig and developed, first we developed a kidney transplant model in pig, and we tested this technology. So what actually happened is that we took the recipient pig, And 14 days before transplant, we preconditioned the recipient with cells treated with mitomycin coming from the organ donor. And then we transplanted the kidney, and then we waited to see what's happening. But before this, we had to develop the allogeneic kidney transplant model in pigs. And for this, it was Professor Matos which did, together with us, this extraordinary study, which actually, from the anatomical point of view, prepared us and made us very aware that the anatomy of the renal hilum and the kidneys in pig is very different from one pig to the other, and prepared us for the technique that we were about to describe. For example, this here, you see that, for example, the left kidney has multiple renal arteries and so on and so forth. This was actually very important for as a preliminary study for our transplantation. Then we went into animals, and then we dissected the kidneys and we transplanted them in a heterotopic manner. And I was telling you about the variation of the anatomy. We have found an atypic kidney, which actually was transplanted very successfully, but this was one kidney that was supranumerary and was very low with the renal artery on the iliac vessels. This is the kidney harvested, and this is the kidney successfully transplanted, revascularized. Of course, as I told you, this was a collective effort, and I have to mention here the group from Professor Ionel Popescu from Daniel Institute. Of course, Dr. Hoinoiu, Associate Professor Joachim Belegan from Lucian with his team, our dear anesthetic Dr. Natsu, my colleague Sorin Barat, and so on and so forth. This was published, and regarding the urinary drainage, we actually had two models. One model in which we transplanted an additional kidney and drained the urine inside the bladder, and one model in which we drained it out. This we left alone because it was very difficult to care for these animals. So this was actually the study design. Sorry. Just want to explain the study design very good so that everybody can understand how we follow this. So at day -14, we preconditioned the recipient with cyclosporine in order for them to accept the cells coming from the donor, which we transplanted in day 7. And then we withdraw the cyclosporine, the immunosuppressant, from the future to-be organ recipients. And this is a graphic showing the plasmatic concentration of cyclosporine. And this was before transplantation, and this was at the preconditioning. So before transplantation, we had very low levels of cyclosporine, so we could not get false positive results. And then we got transplantation. We followed up the transplants by true cut biopsies every 48 hours, and using the BAMF2 criteria, which is a classic criteria for kidney, to evaluate the rejection during kidney transplantation. And we used the ultrasound measurement of the peak systolic velocity at the level of the allograft renal artery. And this is the classification, so below 5 meters per second, you can consider this being full rejection. And these are the results we obtained. So regarding the peak systolic velocity, you can see here that the animals which were only treated with cells, not treated with mitomycin, those curves showed that the rejection was happening very early in the process. While having the animals which were preconditioned with donor-derived mitomycin C-treated lymphocytes, they were rejecting their grafts significantly later. Regarding the histopathological aspect, there was a very clear difference in between groups, and this is the group which received only donor-derived cells. You can see here the pattern of antibody-mediated rejection. So this is the pattern of hyperacute rejection. The entire structure of the microscopic structure of the kidney being destroyed, infiltrated with blood clots and cells. This is the native. This is an animal which did not receive anything, only an allogeneic kidney transplant. You can see that the structures of the nephrons are still intact, and this is the mitomycin C-treated group, where you can see here a very mild rejection happening on some parts of the kidney with the microscopic structure of the tissue still preserved at the time when we declared rejection. So we have closed the gap successfully in between mice and human, and all these results are now, I have the pleasure to tell you that all the results have been submitted and were successfully accepted and are awaiting publication in Science Transformational Medicine. This is a journal where the impact factor is about 10% and so on. The last point, what I want to tell you about mitomycin is that we have started from one cell, then took a drug, then combined the cell with this drug, and then tested this entire strategy until we have found an effect in humans. And now the University of Heidelberg has actually 2 planned phase 1 clinical trials. First, to treat relapsing disease in multiple sclerosis patients which are refractory to any kind of medicine, and to prevent kidney allograft rejection in patients receiving this type of treatment in that medical center. Regarding therapeutic angiogenesis, this was again a combined effort together with the vascular surgery department where I had the honor to work in these years, and the Center of Immunophysiology and Biotechnologies at Professor Ionescu. And what we have actually, why we have actually started